Okay, so is my screen is shared with all of you?
Yes, I think your screen is visible to us, but could you please try to see if we can share the
Slide.
PowerPoint? Yes, please.
Is it now?
Yes, it is. Could you please make it full screen?
Okay, full screen. Okay. Actually, my system is a bit slow, so it will take a little bit time, like 1 or 2 minutes. Yes.
That's
Is it
perfect.
appearing?
We are also slow a little bit. Yes, I think it's visible.
Okay. So the slide transition is taking place. Oh, okay.
Yes, it's going on, yeah.
So should I? We should wait, right, for some other students to join.
Dr. Saroj Ghosh? Sir? Okay.
Good
So
evening.
on
Good
behalf
evening.
of Zoological Association of Burdwan, myself, Dr. Sandas Bandyopadhyay, and Dr. Saroj Ghosh are present. So should we wait for a little more, 1 or 2 minutes more, or should we start, Dr. Ghosh? Okay, then.
Okay.
So I think we'll start with introducing our speaker today, and our people are still joining. So, all right.
Okay,
Okay,
let's
so
wait for 1 or 2 minutes. If more students need to join,
no, I think they.
no problem.
I think they'll keep joining. So
Okay, okay.
yes, yes. Okay, so let us just go ahead. So all right. So hello everyone. This evening, the task of introducing our guest of honor and speaker is a great privilege. I feel elated to introduce Dr. Junaid Jibran Javed for this session organized by the Zoological Association of Burdwan. This evening, we have Dr. Javed, an immunologist by profession and currently serving as an assistant professor in the School of Biotechnology, Presidency University, Kolkata. Dr. Javed, after passing his bachelor degree from Maulana Azad College, Kolkata, completed his master's from University of Kolkata, followed by which he pursued the doctoral degree from the Bose Institute, also in Kolkata. Dr. Javed has authored and co-authored numerous publications and peer-reviewed journals, which are of international repute. We'd also like to convey a sincere thanks to all the attendees, including the students who are making this session successful. Dr. Junaid Jibran Javed will speak on the amino acid structure and classification. Let's welcome him and begin with the session. Dr. Javed, please continue.
Thank you very much, Dr. Bandyopadhyay. It's really a privilege to teach students of undergraduation, and thank you very much for asking me to share some teaching experience with these students. So I hope by now many of the students have joined. So I'm just starting my session right now. I think the slide is visible and the full screen is also visible. Oh, sorry. And the full screen mode is also visible to all of you. Am I audible?
Yes, you are.
Okay. So a very good evening, everyone. As you know that the topic which I'm going to discuss today is actually the amino acid, the basic structure of amino acid, the classification, the physiological and electrochemical property of amino acid, and the physiological role of this amino acid as a whole. So let's see how far we can go with this time slot. So the entire topic is like the amino acid. We need to understand the structure and how the amino acid is actually grouped under the different categories like polar, non-polar with acidic side chain, like amine group side chain, and so on. And after that, we will discuss about the general and electrochemical properties of amino acid, where we will discuss about this Zwitterion and the isoelectric point and how to calculate this isoelectric point for different amino acid, and then we will move ahead with this physiological importance of essential and non-essential amino acid. So let's begin today's topic with the little bit of basic of this amino acid structure. So you know that our body is made up of various biomolecules. These biomolecules are like protein, these biomolecules are like carbohydrate, these biomolecules are nucleic acid, and these biomolecules are lipids. Okay, so among these four categories, if you want to define each biomolecule, there is a specific definition for every biomolecules. Like if you want to define nucleic acid, then these are the polymer of nucleotides. What is nucleotide? Then we have to understand this adenine, guanine, cytosine, thymine, so on and so forth. Likewise, if you want to discuss and know about carbohydrate, then we have to have a particular definition for this carbohydrate. Like the carbohydrates are the molecule which contain polyhydroxyaldehyde, then polyhydroxyketone. Then there is some monomeric form of carbohydrate where we cannot go further breaking down, where we cannot go by breaking down it further. Likewise, if you have lipid or fat, we have certain definition. So in case of protein and amino acid, these biomolecules have a specific definition also. So amino acid is the organic acid compounds and the building block of the very important biomolecule. These are called proteins. Okay, basically this amino acid is made up of a very common component of the organic chemistry, that is carbon, nitrogen, oxygen and hydrogen. Apart from that, in certain other amino acid, it has been found that they contain sulfur. So I will come towards those particular amino acid and how the structure and it can be grouped under different categories, that I will also discuss about today. So these amino acids, as you know, this is the building block of proteins. Okay, so protein, proteins are the very essential biomolecule of our body. The muscles, the actin and the myosin is made up of protein. The rhodopsin, the receptors in our eyes are made up of protein. The spider silk, silk industry on which it is running, it is made up of proteins. Likewise, the ferritin, which actually conjugate with iron, it is made up of protein. The rubisco enzyme, which is present, very much present in the plant cells, these are made up of protein. The hemoglobin, the heme is the globin part of the heme is actually a protein. The immunoglobulin, all these category of antibodies is nothing but basically are protein. And this insulin, this is a very important hormone. I hope that every one of you know about this insulin, that this the diabetes is a very common problem nowadays. Insulin resistance and this kind of problem is very common. So the proteins are anything, like protein can be functional protein, protein can be structural protein. If you want to understand structural protein, then actin and myosin, which makes our muscles, these are structural proteins. If you want to understand about functional protein, then any kind of enzymes like this rubisco, GAPDH, like glyceraldehyde 3-phosphate dehydrogenase, or any enzyme which convert glucose to glucose 6-phosphate. So these are the kind of enzyme which are called functional protein. Okay, so proteins are basically made up of amino acid. If you will break down protein, then you will get some monomers. These monomers are called amino acid. So now the thing is, as the word itself can be defined as amino acid, that means a molecule which contain both the amine group and the acidic group. So the amino acid actually contains three main groups. That is an amine group, a carboxylic group, and the R side chain group. Okay, so this is the basic representation of the amino acid. Amino acid contain an amine group NH2 or NH3 plus in most protonated state. I will talk about it in a greater detail when I will come across this electrochemical properties of this amino acid. It contains this carboxylic group, so that's why it is called amino acid. Apart from that, as you know that carbon has four hand, so four valency. So in this four hand, one hand contains this amino group, one hand contains this carboxylic group, one hand contains this hydrogen, and the another one is containing this R group. This R is not a common alkyl group what we discuss in organic chemistry. This is actually a particular kind of side chain that gives a particular name to different amino acid. Okay, so if you want to learn more about this different grouping, you can see this chart. This particular slide is showing how many types of amino acids are there and how they can be grouped. This is very important, that these things you need to learn and you have to memorize these things. In biology, some things you need to memorize, something you need to understand. Okay, so these things are you need to memorize, like the structures. For example, this glycine, if you want to talk about this glycine, then you can see this is the simplest amino acid. This contains this carboxylic group, it contains this amino group, it contains this R side chain and hydrogen, and the R side chain is only hydrogen. So glycine contain H as a R side chain. Now the glycine, every amino acid, the nomenclature of amino acid is like full name glycine, then it has a triple abbreviated name like GLY stands for glycine, and a single abbreviated name, that is G stands for glycine. Okay, so this is not common for all, like glycine starts with G, so it is somehow it is G, but in case of lysine, let me check. Okay, this lysine starts with L, but its single alphabet code is K. Okay, so, like arginine starting with A, but its single alphabet code is R. So likewise, for this 20 amino acid, this is the task of you student, that you need to understand which amino acid is coded by which 3-letter abbreviation and which 1-letter abbreviation. Okay, now let's see about the structure, because the amino acid structure itself has a big biochemistry in it. For example, if you come from glycine to alanine, you can see that the glycine has H group as a R side chain, but in case of alanine, this H becomes CH3. So the difference is like only hydrogen if it is there in the R group. R means this particular side chains we are referring it as R. So if in this hand it is H, this is glycine. If it in this hand it is CH3, then it is alanine. Now likewise, if you will keep on going like CH plus CH3, then it is valine. Then if you will come, I will talk about this later, but from valine then come to leucine and isoleucine. So again this aliphatic chain, that is long open chain of the carbon, is continuing. Here 3 carbon were there, here 4 carbon is there, and here 5 carbon, 1 2 3, sorry, here 1 2 3 4, here 1 2 3 4. Okay, so this is leucine and this is isoleucine. So iso means another the same chemical formula but different structural formula. So these 4 carbon chain is there, but the location of this carbon is different, so that's why this is called iso of this leucine, so isoleucine. Okay, so likewise this is just the aliphatic carbon chain is increasing, so different name of this amino acid and with different chemical and physical properties. Now some amino acid you will see that it contains sulfur side chains like cysteine and methionine, it contains sulfur SH group. Okay, so these two are the amino acid which is very much known for their sulfur containing group, and these amino acids are very much important in bacteriological system, because this heavy metal like silver, like gold, it actually binds with this sulfhydryl group of these amino acid and make the biomolecule inactive, and this is the way through which we actually target bacterial cell. The antimicrobial agent has been designed in such a way. Whether the protein contain mole sulfhydryl group, then we will attack it with the help of some heavy metals. Okay, so every amino acid structure itself has some more biochemistry in it. Okay, so like some amino acid has this organic structure, aromatic structure, close chain structure like tryptophan and phenylalanine. Some amino acid are polar in nature. Polar in nature means these are amino acid which does not contain any charge side group, charge R group. But here we can see that it is containing OH group. which is negatively charged. It contains OH group. So serine and threonine are the two amino acids which contain OH side chain. Okay, so these are the negative charge amino acids. Again, if you'll come to tyrosine, again it contains OH side chain with aromatic group. So this is another polar amino acid. If we'll talk about this asparagine and glutamine, so asparagine and glutamine has a property. Before that, let's discuss this aspartic acid and glutamic acid. So aspartic acid and glutamic acid are the two amino acid which is acidic amino acid. So always remember, when we will talk about amino acid, these 20 amino acid structure with its triple alphabet name, with its single code name, we should reflect in our mind. This is very important. If you will go for the higher studies, their structure, their function, everything is actually relatable when you will work. So this negatively charged amino acid is aspartic acid and glutamic acid. You can see that in the R side group, it contains CH3, CH2 and COO, and here CH2, CH2 and CO. So these are called aspartic acid and glutamic acid because these amino acid contain the acidic side group. Now if this acidic side group of the aspartic acid is combined with NH2, so instead of O, we are replacing it with amine side group, then it will be CONH2. So this CONH2 becomes, from aspartic acid, it is becoming asparagine. So the name is also different. This is ASP and this is ASN. And the single code for aspartic acid is D and the single code for asparagine is N. Okay. Likewise, if you talk about this glutamic acid, then the glutamic acid has C double bond O N side chain. But if you replace this O with NH2, then the glutamic acid becomes glutamine. So this is how we actually relate one amino acid with another amino acid in order to remember it more perfectly. Okay, that's why I'm taking more time for this slide to explain you. Okay, so some amino acids are positively charged amino acid, like this lysine, arginine, and histidines. Okay, so these are very much important amino acid because these are the amino acid which actually the reserve of positively charged amino group. And when another amino acid needs some kind of amine side chain, then these along with some glutamine becomes the source of this amine group for another amino acids. Okay, so these are the amino acid which contain the positive charge side chains, like lysine, like arginine. It contain 1, 2, 3 amine group. Like histidine contain 1, 2 amino group along with this. This is one of the integral part of this aromatic cyclic ring. So this is the structure of amino acid. The amino acid can be classified in non-polar, polar group, positive charge side chain group, and that is basic amino acid, and the negative side chain group, that is the acidic amino acid. Apart from that, two amino acids are the sulfur containing amino acid, which are called cysteine and methionine, and two amino acids are OH containing amino acid. In fact, three amino acids are OH containing amino acid, which is called serine, threonine, and this tyrosine. Okay, so this is all about the 20 different types of amino acid and their different types of structures. Now the thing is, this is true that we have 20 different types of amino acid, but the reality is that we have more than 100 amino acid. But why we actually talk about this 20 amino acid? Because these 20 amino acid are called standard amino acid, which has been coded by the genetic code. As I hope that all of you are well aware of this genetic code, that we have this triplet genetic code. Each genetic code is specific for a particular kind of amino acid. So all these codes are for the standard amino acid, and these are the list of standard amino acid. But apart from that, some amino acids are also there which are not coded by these kind of codes. So these are called non-standard amino acid. For example, the the non-standard, but it is present in the protein. These are amino acid are not required to build protein, have a vital role as metabolic intermediate. For example, hydroxyproline, hydroxylysine, carboxyl glutamate, diamino pimelated. So these are the some of the amino acid which are non-standard amino acid. Some are non-standard amino acid but non-protein amino acid. They are not also present in protein, but these are considered as amino acid because of their structural properties. Like they have amine group, they have acidic group, and somewhat they behave like the amino acid behave. So these are the derivative of amino acid and have role in metabolism. For example, this butyrate, alpha aminobutyrate, citrulline, ornithine, beta alanine. So these are the other amino acid which are considered as non-standard amino acid apart from those 20 standard amino acid. Okay, so I'm just frequently changing the slides. So if any of you have any doubt, please stop me, please ask me meanwhile of my lecture. Okay, no problem. Moving ahead to the next thing, that is what is the properties of alpha amino acid and why the name is such like that, alpha amino acid. Okay, so the common property of all proteins is that they consist of long chain of alpha amino acid. The alpha amino acid are so called because the alpha carbon atom in the molecule carries an amino group and the acidic group. So in organic chemistry we have a rule that if the functional group like COOH is attached to the carbon, then this carbon is known as the first carbon to which the functional group is attached is called alpha carbon. Subsequently, the next carbon will be called beta carbon, and then gamma carbon, and then so on and so forth is continuous. So if any group is attached to this alpha carbon, then it is called alpha amino. If it is alpha hydroxy, then alpha hydroxy. So that's why the amino acid, these amino acid is known as alpha amino acid. Why? Because the alpha carbon, the first carbon to this functional group COOH is alpha carbon, and the amino acid is also attached to this alpha carbon. That's why it is known as alpha amino acid. Now the thing is that you will see whenever we talk about some proteins, we talk about the N terminal of the protein, we talk about the C terminal of the protein. So what exactly that means? The thing is that when two amino acid combine, like this one amino acid, then another amino acid. So the first carboxyl group of the amino acid combines with the amino group of the another amino acid. It will form peptide bond. The peptide bond is nothing but CONH group. This CO and this NH group, and this OH plus H becomes water, and they actually come out of this peptide bond. So this is actually the peptide bond, CONH group. So when the peptide bond form, this is called dipeptide. Why? Because this contains amino acid 1, then amino acid 2. So this is a dipeptide. Subsequently, if you will add another amino acid, it becomes tripeptide. So peptide means what? They contain several number of amino acids. Okay. Now the peptide, you can see that at one terminal there the peptide has n terminal end. That means this N terminal is non-conjugated to any of this carboxylic acid group of the another amino acid. And in the another terminal, it has a carboxylic acid group. That's why every protein will start from this N terminal and end with C terminal. Meanwhile, it has lots of bonding, lots of stabilizing forces like hydrophobic interaction, electrostatic interaction, van der Waals force, hydrogen bonding, and so on and so forth. So these kind of things come into play and we get a functional protein. So the difference between a peptide and a functional protein is that the functional protein is stabilized under the three-dimensional conformation changes, but the peptide is these are the bonding of several numbers of amino acid. So ultimately, this is how we actually name the N terminal, the C terminal. Okay, like you can see that this is the tripeptide. There is one CONH, this is another CONH. So two peptide bonds means 1, 2, 3 amino acid is attached. Okay, so tripeptide, you can see that here is one amino terminal, so N terminal, and the one C terminal is vacant. This is not occupied with another side chain. So likewise, if it is a long, very long amino acid chain which is a part of the protein, then always it has a N terminal and the C terminal. So this is how we actually relate that what N terminal means, what C terminal means, and how the peptide bond formation take place. So if this is a polypeptide, and once you will let the polypeptide to be stabilized under the equilibrium condition, that is a proper pH, proper temperature, then it will form some conformational changes. It will appear as a three-dimensional object. Then only it will be a functional protein. So always remember, a protein is functional when it has a proper conformation. If you will disturb the conformation, if any of the bond of the protein will be disturbed, then the protein will not be functional, whether it is structural protein, whether it's functional protein. So that's why a protein is functional under its own ambience, like it has its own pH, it has its own temperature, likewise. Okay, so and even if it is stable, even if it has a very well established three-dimensional conformation, still it will be showing this N terminal end and the C terminal end, because the N terminal will always be free and the C terminal will always be freely occupied. Now coming, I hope till now I'm able to explain you the basic stuff related to this amino acid. Moving ahead. Okay, so this is the N terminal and this is the C terminal. Moving ahead with the general properties of this amino acid. So you know that we have, when we talk about amino acid, the only thing comes to our mind is that 20 standard amino acid. Okay, we are not talking about the non-standard amino acid which contain hydroxyl proline or selenocysteine or ornithine or citrulline and so on. We are just talking about this 20 standard amino acid which has been coded by the genetic code and which is a part of our normal protein. So every amino acid, you know that due to the presence of this R side chain, the amino acid actually named differently. Glycine has H as a R side chain, alanine has CH3 as a R side chain. So for 20 different amino acid, we have 20 different R side chain, and for each amino acid there is different in physical and chemical properties, like the color, taste, solubility, polarity and titration value of each amino acid is different from another. There is a specific chart that will give you the idea which amino acid is, how it actually looks like. If you will get the opportunity to perform thin layer chromatography experiment, you will see that every amino acid, based on their side chain, their motility is different in the TLC plate, and you will quantify if there is some formula to understand which dot is which amino acid with the help of the RF value. Okay, so this RF value actually comes when you will see the movement of this amino acid. So this is actually a TLC, thin layer chromatography experiment, through which we actually identify different types of amino acid. So these are the physical property. For the chemical property, since you know that amino acid contains the COH group, amino acid contains this NH2 group, and amino acid contains this both COH and NH2 group. Apart from this normal alpha COH and alpha amino group, like aspartic acid and glutamic acid contain extra COH group, like lysine and arginine contain extra amino group. So due to this R chain also, so an amino acid structure and amino acid's property is actually the reflection of the different properties of their side chains. Like if arginine is a basic amino acid because it has lots of amine group in its side chain. Aspartic acid is an acidic amino acid because it has COH, a carboxylic acid group in its side chain. Okay, so likewise, we actually define every amino acid differently based on their structure of the different side chain. Now, the important aspect that as an undergraduate student you need to understand is the electrochemical property of the amino acid. Okay, so if we will talk about the electrochemical properties of the amino acid, we have to understand what is Zwitterion and what is isoelectric point. These are the very important thing. Why we are talking, why we focus on this Zwitterion and isoelectric point? Because you know that suppose one fine day you are working with some novel protein, you want to to decode the numbers and the nature of the amino acid with the help of some parameters. So how you will understand that which this protein is made up of which particular type of amino acid. For that, every amino acid should have some particular property. Isoelectric point is one of the that kind of property which actually define a particular amino acid. The glycine has different isoelectric point, like lysine has different isoelectric point. Likewise, aspartic acid has different isoelectric point. So this is one of the parameter with the help of which we can understand the identity of a particular amino acid, and we can utilize it even in the formation of the peptide. Suppose you are working with some drug delivery system and you wanted to synthesize a particular kind of peptide with the help of 4 amino acid, and that peptide will exactly behave like an antibiotic. So in that case, you need to understand the isoelectric point of each amino acid. Then you can add it, then you can make a functional peptide, and then you can work with that particular drug, improved drug delivery system. Okay, so that's why as a biochemist, as a particular person engaged with amino acid study, the electrochemical properties and this Zwitterion and isoelectric point is very important. Now let's see how actually we understand, or how what are these Zwitterion and how what are these isoelectric point. So in chemistry, a Zwitterion, also called a dipolar ion, is a negatively charged functional group. For example, if I will talk about hydrochloric acid, so you know that hydrochloric acid under ionic condition, it will give you only polar group. Like it will have only, suppose if I'm talking about some molecule like acid, HCOOH, it will give you the COH group. Okay, likewise, if some molecule organic compound is I'm talking about, so it will only non-polar nature it will show. But some molecules are there which will have both positive as well as negative charge in its structure. For example, just look at this. This is a particular amino acid contain carboxylic group, the amino group, this R side chain, and this hydrogen group, because of the 4 different valency of the carbon. Now you can see that at this stage, that is under very low pH condition. So the pH value, you know that it is actually the reflection of the protonation. So highly protonated means highly dominant positive state will be reflected when the molecule is under very low pH, in the acidic pH. Okay, so this is actually showing the very highly protonated state of the amino acid. Here you are getting only +1 charge of this total group, but when you will rise the pH gradually, there will be deprotonation from this place, from this carboxylic acid group. So it will be ionized as COO minus. Now the total entire charge of this amino acid group, this amino acid is +1 and -1. So ultimately it is 0. So this is the state where the amino acid movement towards anode and the cathode will not be there. It will be stabilized under the normal equilibrium, and there is no movement at all in this amino acid. This particular state of the amino acid is called Zwitterion. As I've told you, a molecule can exist either in totally negative state or it can exist in totally positive state. But this amino acid has a property due to the presence of this carboxylic group as well as amino group. It can exist in both state, the electronegative and the electropositive. So this kind of state of the molecule is called Zwitterion state. Okay, so in this Zwitterion state, the net charge of the molecule is 0. There is no net movement of the molecule. Again, if you will keep on rising the pH, then another proton will be released from, because this is the highly protonated group. So another proton will be released from this group. So ultimately it will be neutralized. So here the total scenario is that the molecule contains 1 negative charge. So from here, this is +1. then we are coming to 0, then we are going to -1. So gradually the charge is decreasing, like the nature of charge is getting converted from highly positive towards highly negative through intermediate of 0. So this intermediate is the Zwitterion state. Why it is important? Because every uh, among these 20 amino acid, each amino acid has a different pH for its Zwitterion state, and that different pH actually give you the isoelectric point, the point at which the amino acid does not have any movement, and this isoelectric point is one of the parameter for the identification of this amino acid. So which amino acid, every amino acid has a fixed pi value. So what is this isoelectric point? Just let me discuss. At a certain pH, isoelectric point is nothing but a pH value specific for each amino acid. The amino acid can exist in the dipolar form, fully ionized but with no electric charge. This is fully ionized. Here we have negative charge, here we have positive charge, but still the entire charge of the molecule is actually 0. So this is, although the molecule is displaying the full charge of the capacity it has, but the entire charge is negative plus positive, so it is 0. So this point is called isoelectric. So as you know, the name itself suggests isoelectric means equally electrical point. The characteristic pH at which the net electric charge is 0 is called the isoelectric point. So the Zwitterion form is called the isoelectric point. The amino acid at its isoelectric point is called the Zwitterion and is the electrically neutral, not migrating in the electric field. So that's why I wanted to focus more in this particular topic, because this is the thing that we need to understand with respect to the electrochemical property of the amino acid. If you want to decode any amino acid with its nature, then isoelectric point is one of the very important point that we need to understand. Okay, so let's see how we can understand or how we can calculate the pi value of this Zwitterion. Okay, with respect to your further competitive examinations also, these things are very important, that you need to calculate the pi value of certain amino acid. So that you need to learn. Okay, so let's start with the first one. The thing is that because of the lack of this board facility, which I'm not having right now, and I don't know how to work with this touch screen thing because I lack the stylus even. So that's why I have taken this as a screenshot of some videos. Okay, so let me explain you that in case of a 3 amino acid, I will show you gradually examples of 4 amino acid and how this amino acid is being protonated gradually in the different pH, and how to calculate their pi values, isoelectric point. Okay, so the first one is the glycine. You can see that this structure is glycine. The carbon is attached to COOH, the amino group, the H, and the R side is also H. So this is the glycine. Okay. The pH at which the COOH group of the glycine will be protonated is pH 2.3. So lowest pH at which the COH group will be deprotonated. The next is 9.6. You know that there is something called alkaline pH or acidic pH. So this is acidic group, so it will be deprotonated at the acidic pH only. If this is an alkaline group or basic group, this will be deprotonated or protonated at the highest pH, that is alkaline pH. Okay, so step by step, just try to learn this thing. It is showing that it is named the pH value at which the COOH will be neutralized is called, or deprotonated, the H will be removed, is pH 2.3. This is called pKa. pKa is nothing but the first pH at which the COOH group will be ionized. Then another pH at which this NH2 will be ionized is called pK2. Okay, so pKa1 or pKa2 is nothing but two different pH at which the acidic group and the basic group will be protonated or deprotonated. Okay, let's see the movement or the behavior of this glycine molecule. Really, from 1 to we are increasing the pH up to 2.3. So up to 2.3, this is the zone where the amino acid glycine will show the highly protonated state. After pH 2.3 value, gradually the COH group will release one proton. So it will be CO minus. Below that it will have highly protonated, so that's to contain one H. So the total charge of this molecule is +1. Now gradually you are coming beyond this 2.3 pKa value where the OH, this H group will be deprotonated. So now it is -1 and this is +1. So from 2.3 to 9.6, because 9.6 is the another pH value of this deprotonation, up to this junction the molecule will behave as a Zwitterion. Why? Because it has one negative charge, one positive charge. So the Zwitterion will be existing in between the two frame of pH 2.3 and pH 9.6. After pH 9.6, what happened? This protonated NH3 group will again release one H plus and it will become NH2 from NH3 to NH2. Now the entire charge is only -1. So you can see that here it was +1, here it is 0, and here it is -1. Now the thing is how to calculate the PI value, the isoelectric point of this glycine. To calculate the PI value of the glycine, the pKa1, that is this one, plus means the where the Zwitterion actually existing, the the left side and the right side pKa values we need to average. Okay, so the pKa is 2.3, the pKa2 is 9.6. If you will average it, 3 plus 9.6 divided by 2, it will come like 5.95. So that means the glycine will be identified by the molecule which has a PI value of 5.96. At pH 5.95, sorry, it will act as a Zwitterion. So this is the identity of the glycine. Moving to the next amino acid, that is lysine. So meanwhile, I'm again telling you that if you have any question, just stop me and ask me, okay? Because these are the things that you need to understand, and this is how we actually understand the isoelectric point of each amino acid. So okay, so glycine, the structure was very simple: COH here, NH2 here, H and H. Okay, now we are changing this H group to some another R group in lysine. So here the NH3 will be added. So altogether we have three charge group. One is COH, one is NH2, there another NH2. Now the pKa value is pKa1 2.18, pKa2, the another first proton will be liberated from the COH group, the second proton will be liberated from the alpha amino group, and the third proton will be liberated by any other amino group. Okay, so this is the actually criteria. If it is acidic group, then what happened? The first proton will be liberated from the alpha carbon group, the second alpha carboxylic group, the second proton will be liberated from the another COOH group, and the third proton will be liberated from the another amino group or alpha amino group if it is present. Okay, so that means it is present. Okay, so there are two things. So let's see the behavior of lysine under the increasing pH. So as you know that 2.18 is one junction below which the lysine exists as a highly protonated. So this is COOH, this is NH3 plus, this is NH3 plus. So total the charge is +2. Now increasing coming out of 2.18, above 2.18 to next is 8.93. Up to this junction, the proton will be deprotonated in COH and it will be COO minus. So 1 minus, 1 plus, 1 plus. So altogether 2 plus, 1 minus, that means the entire molecule will behave as +1. Still it is not a Zwitterion. The criteria for the Zwitterion is the entire charge will be 0. Let's see next. If you'll go beyond 8.95, this is 8.95. So beyond F.95, then this proton will be liberated. So here we are getting, this is 0, this is NH3 plus, and this is CO minus. So altogether 1 plus and 1 minus, this molecule is displaying net charge is 0. So this is the Zwitterion state. And then if you will move further, then the another proton will be liberated from this one. So it will be NH2. This is NH2, this is CO minus. So ultimately from plus 2, plus 1, 0, then minus 1. So ultimately the entire highly positive state is converting to entire highly negative state. Meanwhile, we are getting a Zwitterion value. Now how to calculate the PI, isoelectric point, for this lysine? For the isoelectric point of the lysine, the criteria is same, that the Zwitterion store, at the Zwitterion step, the left side and the right side, these two pKa values will be considered. So 8.95 plus 10.5 divided by 2, this will give you the isoelectric point for this lysine. Okay, so this is the example of the basic amino acid and how we can find this PI value of this basic amino acid. Okay, so moving ahead with another example, that is a negative charge amino, a negative charge side chain. Like for this tyrosine, OH is negatively charged. How to calculate the PI value and how the behavior will be monitored. Okay, so see, here what happened, the molecule tyrosine in there is the pK value is 2.2, 9.1 and then 10.5. Okay, so since it is not a carboxylic acid group, that's why this will be, the proton will be liberated from here at the very last step. Okay, if it is COH, then after just this carboxylic group, it will be the second one and this will be the third one to liberate the proton. So understand the criteria. These are some biochemical criteria that on the basis of which the proton will be liberated. Okay, so at the very beginning, that is below the pH of 2.2, what happens, the molecule tyrosine will exist as a highly protonated form, that is COH, NH3 plus, and this is OH. So altogether H plus NH3 plus, that means the molecule is showing plus 1 charge coming to above 2.2 value. So the next proton will be liberated from COOH group. So it will be like CO minus, NH3 plus, and this OH is still 0. So ultimately minus and 1 positive charge. So the entire molecule will display the value 0. So this is the Zwitterion state. Let's see the another behavior, because we have 9.1 pH also and this 10.5 pH. This amino group will liberate one proton and it will be neutralized. So, and this is already neutral. So this entire molecule will display -1 electrostatic state. And if you will move ahead further with pH 10.5, then it will be like, here it is already -1, here the proton is also liberated. So from here the proton will be liberated and it will be -1 again. So this -1 and this -1 together it is -2. So now see the movement, like it is +1, then 0, then -1, then -2. So the Zwitterion state is this. If you want to calculate the PI value, then you need to calculate on the basis of the left side value and the right side value. So the left side value was 2.2 and the right side value was 9.1. So average of these two values will give you the PI value of this tyrosine. So sorry, so the PI value of tyrosine is 2.2 plus 9.1 divided by 2, it will be 5.6. So every PI value, like tyrosine has this PI value, lysine has another PI value, aspartic acid has other PI value. So like this we actually understand, okay, which amino acid has which PI value, and this is will help you to, for various aspects, to understand the protein structure, to understand the protein function, which pH will be suitable for this protein function or not. Okay. Likewise, this is an acidic amino acid. So in this case, what happened? The very low pH, it will be highly protonated. The first deprotonation will be happen from the alpha carboxylic group. The second deprotonation will take place from the another carboxylic group, and the third deprotonation will take place from the alpha amino group. So here the Zwitterion state is this one. So the PI value will be calculated from 1.88 and then 3.65. The average of this thing will give you the PI value of this particular amino acid. Okay, so this is how we actually calculate the PI value. This is what we actually know about the Zwitterion for the 20 different amino acids. This is not possible to show you the Zwitterion and the PI value. So I'm just summarizing this up to here. Okay, now the thing is the physiological importance of this essential amino acid. I have just one or two slide more. Okay, so the physiological importance of this essential and non-essential amino acid, like you know that what is this essential and non-essential amino acid. So essential amino acid which cannot be made by our body. So anything you cannot produce in your body but you need it, this is become essential. This is how we actually judge the criteria for essentiality of a molecule. This is same for this fatty acid also and some other molecules also. So these actually will be produced, this actually we have to take from some food sources. So what are the different essential amino acid? These arginine, histidine, isoleucine, leucine, lysine, methionine, these are the essential amino acid. Then what are non-essential amino acids? So apart from this 10 or 8 amino acids, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this 10 amino acid, another 10, actually among this 10 amino acid, 8 amino acid is actually essential for all infant and adult, but 2 is actually in case of the infant it is more essential. So like this. So apart from this 10 amino acid, the another 10 amino acid comes under the non-essential because the body can produce it. We have the pathway for the synthesis of this amino acid like asparagine, aspartic acid, alanine, glutamic acid, glutamine and all. So these are the group of essential and non-essential amino acid. Non-essential does not mean we don't need it. We need it, but we have the ability to produce it. So we don't have to rely upon the food sources or anything else. Okay. These are the list of food from where we get this essential amino acid like egg, parmesan, sesame, soy protein. Okay. So various actually what we say is what the non-vegetarian foods like fish, meat, poultries, egg, these actually contains the rich amount of the essential amino acid. That's why their protein is also called first class protein. If you will study about the category of protein, some proteins are called first class protein, some protein are called second class protein. The first class proteins are those protein which contain all these essential or many of these essential amino acids. So basically it has been found that mostly those poultry, egg, meat and fish, they are rich in essential amino acid. That's why their protein is also called first class protein. Okay, so I think I should summarize up to here only. If you have any question, then you can ask me. Thank you very much everyone. Thank you very much organizers also.
Thank you very much, Dr. Junaid Jibran Javed. So I'd like to request the students, if they have any questions, please ask the question to the speaker.
Yes. Yes.
Hello, sir.
When we calculate
All
PI,
right. So yes, please first utter your name and then continue the questions of the.
sir, I am Sotik Saha.
Shobtik. Okay, Shobtik, in which year you are?
I am in second year.
All right. Okay, please ask your question.
Sir, during calculate of PI value,
Mhm.
in case of lysine,
Mhm.
we consider the last two pK value, but
Yes.
during tyrosine,
Mhm.
we consider first two pK value.
Okay. Yes. Yes. Then what's your question? Then why it
So,
is so, right?
yes,
Okay. See, the criteria is that when it is only the carboxylic group and the amino group, then the first proton will be liberated from the alpha carboxylic group. Then the second proton will be liberated from the alpha amino group. But if it is the third amino group, then the third amino group will be the third one to liberate a proton. In case of tyrosine, the third group is not amino group. This is OH group. Okay, so that's why here the criteria is different. first is alpha carboxylic group will liberate, second is alpha amino group will liberate, then the OH group will liberate the proton and it will become O minus. In case of aspartic acid and glutamic acid, it contains acidic amino acid. So in case of acidic amino acid, apart from the alpha carboxylic group, it contains additional carboxylic group. So the carboxylic group are those groups which are acidic in nature. So the first proton will be liberated from the alpha carboxylic group. The second proton will be liberated from another carboxylic group only. Then the third proton will be liberated from the alpha amino group. So these are the criteria on the basis of which we actually understand how the amino acid is behaving under different pH. I hope I have given the answer of your question.
Okay, sir. Okay.
Next question by Pranay Kundu. Please pronounce your full name and the college from where you are, and then please continue the question.
Sir, my name is Pranay Kundu. I am from Vijayanaran Mahavidyalaya, Zoology 3rd semester. Sir, I have the same question, and that is why tyrosine PI value is last 2. It is explained. Okay, thank you, sir.
Okay.
All right, I
Yes.
think you've already got your answer. So do you have any more question?
I think Pranay does not have any more question. Yeah.
No,
Yes.
sir.
So any one of you? No question? All right. So if there's no more question, then I would like to request Dr. Saroj Kumar Ghosh on behalf of Zoological Association Burdwan to express a vote of thanks. Dr. Ghosh, please continue.
On behalf of the organizing committee and the Zoological Association of Burdwan, we express our sincere gratitude to Dr. Javed for accepting our invitation and spend your quality time in favor of undergraduate students of zoology in the online lecture series. We are grateful to you, sir, for your lovely presentation and sharing your rewarding knowledge with us. I think your important lecture on classification and structure of amino acid will be helpful for our students in their educational platform. Many thanks to our dear students for your attendance and taking part in this academic program. With these few words, I conclude the session. Thanks again.
Thank you very much, the participant, and thank you very much, organizers. Thank you very much, Shamla.
Thank you very much.
So I'm leaving now. Thank you.
Yes, please.